Hi, my name is Pamela Kunz, and I'm a medical oncologist at Yale Cancer Center and Yale School of Medicine. It is my pleasure to join you today. I will be speaking about understanding the latest NET clinical trials. These are my disclosures. And this is the brief outline of what we'll be covering. I'll first talk about clinical trial facts and myths, how to interpret clinical trial results, new NET trials, some approaches that are on the horizon in cancer treatments and how we can apply those to NETs, and how you can find a clinical trial. First, why are clinical trials important? Before me, you've already heard about the latest advances in NET treatments. These treatments, in fact, become available because of clinical trials. They are a key research tool for advancing medical knowledge and patient care. And clinical research is done to learn whether a new approach is safe and effective, and which treatments or strategies work best for certain cancers or groups of people. Yet a minority of U.S. adults with cancer participate in clinical trials, only 7%. So let's talk about myths and facts. And I think there's actually a lot of stigma and myths out there about clinical trials. So the first myth is that clinical trial participants are, quote, guinea pigs, when in fact the fact is that strict guidelines are in place to inform clinical trial volunteers and protect their safety. Another myth is that if I join a clinical trial, I will receive a sugar pill or placebo instead of the real medication. Placebos are in fact becoming less commonly used. They are generally only ethical if no standard treatments are available, and you'll find that with some of the newer NET trials, placebos are no longer being used because we have so many more approved therapies. Another myth is that once I decide to participate in a trial, I cannot change my mind. And the fact is that you're free to leave the trial at any time, even after you have signed the informed consent. Myth 4, there are no costs associated with a clinical trial. And the fact is that many aspects of a trial are covered, though there may be some participants who will have copays that are associated with standard items. Myth 5, clinical trials are only for patients without other options. And the fact is that there are many clinical trials available for all stages and many situations. Briefly, I'd like to just cover the path to drug approval and how really every drug starts first in the lab, and this is called the preclinical phase. This includes test tubes and mice. And then the drug is approved to move into humans, and that first phase in humans is called phase 1. It usually involves 20 to 80 participants. The intent of a phase 1 trial is to test safety. Once it passes that bar, and once we determine the maximum tolerated dose or the safe dose, a drug will move into phase 2. This can be randomized or a single-arm study, often 100 to 300 participants. The intent of a phase 2 trial is to test efficacy, usually in a specific cancer like NET. Once it passes that phase 2 bar, it moves into phase 3, testing 1,000 to 3,000 participants. These studies are usually randomized. Once it passes a phase 3 step, it then moves to the FDA for review to confirm safety and effectiveness, and this is a very rigorous review by the FDA. And then after an FDA approval, there are continued studies called phase 4. These are real-world studies to continue following for safety. So in terms of the design and interpretation of clinical trials, so we look at eligibility criteria, which NETs, which grade, which primary site, is the cancer progressing or not, has a patient received prior treatment, and we also look at the measures of success. These are called endpoints for a clinical trial. So these include things like progression-free survival, or how long does it take for the tumor to grow, overall survival, how long do patients live, a response rate, how much does the tumor shrink, and safety. And in clinical trial speak, we call these adverse events. So let's just briefly talk about how we interpret results of clinical trials. So if you are reading a scientific journal, you will see these things called survival curves. This is an example from a trial that I had the opportunity to lead that was the ECOG 2211 trial of capecitabine versus Cape-Tem. This is the progression-free survival curve in yellow. is the progression-free survival curve for CAPE-TEM, and in blue for TEM alone. The horizontal axis along the bottom is in months, and the vertical axis is the y-axis, and that is the progression-free survival probability. That dashed line is the median progression-free survival. And then the separation of the curves between the blue and the yellow is good. With the yellow being much higher than the blue, it means that that yellow arm, or the patients receiving CAP-TEM, had a longer median progression-free survival. So that arm actually did the best in this study. And a hazard ratio means the hazard of progression. And so the p-value and the hazard ratio, low is good for both. So a low p-value means there's statistically significant difference between the two arms, and a low hazard ratio means that there is a lower hazard of progression with that CAPE and TEM. So moving on to tumor measurements, we use the RECIST criteria, or the response evaluation criteria in solid tumors, to measure the size of tumors over time while a patient's on a clinical trial. A lot of patients will ask me, well, why can't you measure every tumor? Well, for a clinical trial, we actually pick representative spots, so a maximum of 5 total lesions, up to 2 per organ. We measure the sum of the longest diameter and measure that over time. And then that percent change is grouped into 4 categories: complete response, partial response, stable disease, or progressive disease. Let's use an example. So imagine that this is a baseline tumor measurement. This is a liver with 2 metastases in yellow at baseline or before treatment. You can see the 2 measurements. I've put them both in that cartoon of the liver and in the table below. So liver lesion number 1 is measured at 2.5 centimeters. Liver lesion number 2 is 6.5. The sum of that is 9 centimeters. After a patient has had 3 months of treatment, we're seeing tumor shrinkage. You can see that we've added an additional column at 3 months. And then you can see that there is a third column at 6 months, and the patient has had continued shrinkage. And so then we sum that up. We measure the percent change from baseline. And so at the 3-month mark, it was 17% shrinkage. And at the 6-month mark, it was 44% shrinkage. And that has met the criteria for partial response. And so this is the example from the ECOG 2211 trial. In the CAP-TEM arm, there was 40% shrinkage, and then the temozolomide arm, there was 34% shrinkage. So both, in fact, actually had a pretty significant amount of tumor shrinkage. However, the CAP-TEM arm had a little bit longer. Adverse events are collected and recorded every visit by the research team. They are graded both in terms of severity on a 5-point scale. You can see the grades listed there. And they are also measured in terms of relatedness to the medications. So unrelated, unlikely, possible, probable, or definite. So also a 5-point scale. A really important point I'd like to make is that patient-reported outcomes, or PROs, are also increasingly used as a way to measure side effects or toxicity in trials. And I think this is a really important way to engage patients and have the patient's voice as part of the trial. At present, we use something called the NCI Common Terminology Criteria for Adverse Events. Again, it's on this 5-point scale. This helps us quantitate or really measure an adverse event. So let's move into some trials to watch. So you have heard about some of these trials already today by Drs. Haftanderson and Dr. Mitra. I'd like to highlight some trials that I think are really exciting. What I didn't go into, and I'll just briefly mention here, is that there are a number of different sponsors for clinical trials. Sponsors are generally the group or organization that is paying for the company or has the intellectual property. So these 2 trials are sponsored by the federal government, or called the National Clinical Trial Network. The S stands for SWOG and the A stands for Alliance. These are really important ways to do studies, especially in rare diseases, because we can open these trials at many sites. So this SWOG 2104 study is the first study to look at postoperative treatment in pancreatic NETs. I think this will be a very important result regardless. At present, we don't use postoperative treatment. And this builds on that ECOG 2211 study and gives patients 4 months of CAP-TEM versus observation. So it's a randomized study. This alliance study is a study of paraganglioma and pheochromocytoma. It's temozolomide versus temozolomide plus olaparib. It's led by Drs. Perez and Del Rivero. You'll note a longer list of radioligand therapy, or RLT, and LACNETS. This is a very hot area for clinical trial research, is really looking at our various types of radioligand therapy across multiple subtypes. So COMPETE and COMPOSE are using lutetium etatrieotide. The COMPETE study has completed accrual. We are awaiting the results, and the COMPOSE study is still accruing. And then the Alliance 021901 study, led by Drs. Hope and Pata, is in LACNETS. So also very exciting because this is a primary site that we don't have a lot of trials for. So we are excited to examine lutetium dotatate versus everolimus in this setting. And the A022001 study is a clinical trial also using CAPTEM, but comparing that to lutetium dotatate in metastatic pancreatic NET. So again, building on prior success from the ECOG 2211 study. So CAPTEM we use a lot, but this is a very practical question to try to answer with this study because we are faced with this frequently of whether to use ludotatate or whether to use CAPTEM. The Action 1 study is using an alpha therapy, so actinium-225 dotatate versus investigator's choice. And then the last one listed here is NET retreat. So asking the question of after someone has had a full course of 4 cycles of ludotatate, can they receive another 2 at time of progression? And I don't want to omit a discussion on our neuroendocrine carcinomas. This is also an active area of research. There's another NCTN, or National Clinical Trial Network study, the SWOG 2012 study, looking at etoposide and platinum, or carboplatin etoposide, many of you have heard of, plus or minus atezolizumab. So atezolizumab is an immunotherapy. This approach has been used in small cell lung cancer. So for the first time, we have a clinical trial in extrapulmonary small or large cell neuroendocrine carcinoma. So very exciting. We're formally studying immunotherapy in this setting. And then I've listed what I think is an important study. It's being conducted by the French NCI. So this is not available to our US patients, but FOLFIRINOX is a combination of 3 drugs, 5-FU, oxaliplatin, and irinotecan, that's used very commonly in other GI cancers. And we're comparing that to carboplatin and etoposide in this study. So again, a very practical question, and one that I hope will help us identify new treatments for future. Lastly, I'd like to end on, can we translate novel approaches to NETs? And these are 2 examples that are not in NETs, so I want to be very clear about that. But I think that it's given me a lot of hope and enthusiasm about ways we can potentially think about novel approaches. So on the left is an example of a new form of immunotherapy. There is a pancreatic cancer vaccine that's being used following surgery. It's just in clinical trials right now, but a vaccine is another way to manipulate the immune system. As many of our listeners may know, immunotherapy or single agent checkpoint inhibitors have not proven effective in neuroendocrine tumors. So we're really looking for other ways of manipulating the immune system to help fight cancers. So that includes things like vaccines, cellular therapies, CAR T cells. So I'm really hoping that some of these will make their way into the NET space. The other is personalized medicine. The idea from this cartoon here is that if we can identify very specific characteristics about a patient and target that we may be more effective at yielding better results. So we have a sort of red, blue, and green characteristics of these patients, and the idea is that we develop both a biomarker that helps us identify that, and then we tailor our treatments towards that result. So one example that has really exploded in terms of research in the last year or so is targeting KRAS. Now, this is a gene that is occasionally seen in some neuroendocrine tumors. We are seeing it in lung and pancreatic cancer, and this was a gene that was long thought to be undruggable. And in fact, in the last year, we have seen new drug development to target KRAS. And so stay tuned. I think that just broadly, I'm hopeful that these principles will be applicable to NETs. So how can you learn about clinical trials? So number one, talk to your oncologist or NET specialist. They should really be your primary guide. Go to your institution or cancer center websites. I've listed some LA local, but I know this is a national meeting, but go to your institution website. There will undoubtedly be ways to look for clinical trials. Clinicaltrials.gov has actually had a facelift this year. It's a little bit more user-friendly. You can put in search terms. So in this example, I put in neuroendocrine tumors. It will come up with many potential options, but there are also ways to filter. And so it's getting a little bit easier to use. And then lastly, a call out to some of our patient support group sites, LACNETS included, where they have also curated lists of clinical trials for NETs. So take-home points. I hope that I have imparted the hope that I have for the future and why clinical trials are part of that hope. We've really had so many major advances in the last 10 years in terms of treatment and imaging. Clinical trials are critical to making that progress. Radioligand therapy, immunotherapy, and personalized therapies are likely to be incorporated in future NET trials. In fact, I think these three categories are really where we're going to see major advances. And a special thank you for any of you who have participated in clinical trials. I think that I know myself and my colleagues have a lot of gratitude for you. You are helping to move the needle forward. So thank you so much. I always love speaking at this conference. It's really my pleasure.